Thank you for giving us the opportunity to speak today and to share a bit of Variant Bio's work, namely how we use mid-pass whole genome sequencing to enable biomedical genomic studies of diverse populations. First of all, let me give you a bit of background about Variant Bio. We were founded about 2.5 years ago. We're headquartered in Seattle, but we work really around the globe, very internationally, and we partner with populations that have unique health traits, particularly geographically isolated and founder populations. And we do that with the goal of discovering novel gene trait associations that we then take into development of novel therapeutics with a focus on common disease. So as we're pursuing this goal of developing novel treatments, we're dedicated to making genomics more equitable and more inclusive. And we're doing so by having benefit sharing programs set up, both for benefit sharing in the short term, for example, capacity and educational efforts, as well as in the long term through revenue and equity sharing. This is really central to our company, and please go take a look at our website if you're interested to learn more. So genetics studies need more diversity. Currently, there is still a very strong bias towards individuals of European ancestry in genomic studies, and even though there's been a bit of an improvement over the last decade, most populations of non-European background are still really poorly represented. And this bias in research also leads to bias in medicine and healthcare, and therefore only furthers existing health disparities. In terms of global genetic diversity, we still have a lot to learn, especially about biomedical trait associations and about disease mechanisms. However, given this underrepresentation, existing genomic reference panels and arrays right now lack the ability to identify population-specific variation. And therefore, one approach one can use is high-coverage whole genome sequencing, but that is still prohibitively expensive, especially for example, for us as a small startup, but also generally just to empower research studies around the world. So there's a need to be more inclusive and improve how research is conducted and how participants profit from it, both directly through benefit sharing, for example, and indirectly through improved representation. At Variant Bio, we employ a method that we call mid-pass sequencing to set it a bit apart from what is typically referred to as low-pass sequencing, which would target 1x coverage or even less. We target 4x coverage for the majority of the cohort, and then about 10% or less of the cohort at 30x high coverage. We then use really standard and openly accessible methods for variant calling and imputation. Specifically, we use the GATK best practices down to joint variant calling, jointly genotyping 30x and 4x data, then run variant site level filtering with VQSR, followed by call level filtering, where we remove low quality genotype calls, which is very important, before running imputation, which then fills in the missing genotypes. Afterwards, we have a call flagging step that basically looks back at the sequence information to see where imputed calls and reads agree or disagree. And that is really also helpful in downstream quality assessment. Given that 4x coverage sequencing is much more cost-effective than 30x, the overall sequencing budget can be, compared to 30x high coverage only, can be 20% or less with this approach. So we've evaluated our approach extensively. We evaluated our approach on a cohort of 1,500 individuals that are not currently well represented in existing reference panels. 100 genomes were sequenced at 30x, and the remainder at 2 to 6 or 7x coverage. We then compared our imputed mid-pass calls to separately available 30x truth data. We did the same also with genotyping array data that we imputed using the 1,000 Genomes reference panel. And comparing these two approaches, we see that at our targeted 4x, we basically outperform the array-based approach at all metrics, from recall precision to non-reference concordance rate, which basically measures our ability to correctly call genotypes. We also see that this approach is robust across different ethnicities within the cohort, and there will be more information on this and more details in a paper that's coming out hopefully very soon. Also important to note, in comparison to the array-based strategy, is that we really outperform across the allele frequency spectrum at 4x. And obviously, with sequencing, we're able to identify also novel population-specific variants that are not contained in 1,000 genomes. We evaluate also what happens if we lower the cohort size, so going down to 250 individuals from the original 1,500, and we see that this is really robust even in this much smaller cohort. So overall, we really have a method that gives us accuracy above 95% across all metrics or higher, and that at a cost of 20% compared to high coverage sequencing. So how does AWS come into play? All of our setup basically is on AWS. It's been great for us because depending on where we are in a project, we may need either a lot of computational resources and then can scale up a lot. And we've frequently had to increase EC2 limits, especially in the early days. And at other times, we may just not need a lot of compute at all. So we don't have any resources sitting idle that we don't need. Apart from that, it just provides so many convenient services and keeps adding to the list. We use many of the services, including different storage solutions from EBS, EFS, S3 for permanent storage, to organizing and regulating our network structures with VPCs. And also we make heavy use of Lambda functions for various jobs, including, for example, tagging of resources, which we found to be really useful for organization and cost and resource monitoring. And of course, we also use, for our genomics analyses, more of the high performance compute solutions. We run Hail on EMR. We use Athena for several applications. And for the pipeline that I described earlier, we run that on AWS Batch. I'll get into some more detail about that afterward. Something that's very important for us for working internationally, worldwide, is just the availability of AWS. So depending on where we're working in the world, there can be local regulations around genetic data, so that it needs to be analyzed within a certain geographical location. And with the different AWS availability zones, that gives us the possibility to do that. Something that's also come in extremely useful here is CloudFormation templates. So I really recommend setting up everything that you can in CloudFormation as early as you can, so that it's, first of all, reproducible and easily changeable. And also, should you ever set up in a different region, that's obviously super helpful. We also have the well-architected pillars from AWS in mind, particularly focusing on security. So our setup on AWS Batch looks as described in this slide. We use this not only for the variant calling and imputation workflows, but also for other pipelines, for population genomics analyses, and occasionally even when we're downloading bigger data sets from public repositories, if that data is not already conveniently located on S3, which makes things a lot easier. But yeah, we use this in many different settings. So the workflow language that we chose is Nextflow. It's very flexible. It lets us use different scripting languages. It's portable, should we ever use it on any other infrastructure. But most importantly, also it has very nice and easy integration with AWS Batch. It provides a lot of useful features, job retries, pipeline resumes, and resource usage reporting. We have also Nextflow Tower set up along with it to monitor our workflows, and that's been really useful. So most of this is relatively automated, but it still has some manual points along the way. So when we have new data, it gets entered into a Aurora database that basically contains all information from study information to analysis output, file locations in S3, and also workflow information. So when we start a new run, we query this database that returns input for the pipeline, and then we grab the correct Nextflow workflow to go with it and stage it to S3, and from there submit an AWS Batch job that first off runs a main Nextflow node that runs on an on-demand instance and basically submits all its workflow child jobs to further AWS Batch instances and grabbing the correct Docker containers from ECR. It's very similar to a setup that's been developed by AWS. One of the things that we decided to do in the beginning, and which has helped us track resources and costs associated with different workflows, was to set up compute environments on a per project basis. So every batch instance and the corresponding EBS drives will be tagged with a study tag, and that helps us allocate resources and monitor resources and cost in the Cost Explorer. Okay, in terms of services that most help us with cost effectiveness, obviously Spot is a big saver and saves 80%, if not more, of cost associated with EC2 compute, and especially for compute and resource intense and parallelizable jobs, Spot is the way to go. We do still use on-demand for certain jobs that are maybe bottlenecks in the pipeline or that are resource light but may run for quite a long time, so we don't want to risk any instance failures. Also, we found really that the AWS Spot Advisor is helpful in picking the right instance types, and sometimes really restricted to specific types that we see have low interruption rates. In the earlier times, 2 years ago or so, we would use even a lot of R3 instances just because those were highly available. But going to the newest instance types, and for the most part, we currently use R5 family instances. We see also much improved performance compared to the older ones, especially for I/O heavy jobs. On the storage side, we do EBS auto-scaling, which means we start off with a really small EBS drive on the batch instances, around 100 gigabytes usually, and then we scale it up dynamically as disk space is needed up to a certain maximum, which we set usually to 3 terabytes. And given this maximum, which would mean provisioning for the maximum space consumption we could anticipate, we just have a really big cost saving on the EBS side here, which makes EBS really the much smaller factor compared to EC2 in the batch setup. Then for long-term storage, we have we use Glacier Deep Archive. We did in the beginning use intelligent tiering for a while, which was also very useful, especially while we were setting things up. But with the type of data and analysis that we do, we move to a more intentional model where we're actively sending data to Deep Archive when we know it's ready for transitioning into that state. To summarize, AWS really provides a very flexible infrastructure, and that makes it even possible for us to do the work that we do worldwide. Our mid-pass genome sequencing approach is well suited for working with underrepresented populations, and it allows also detection of novel population-specific variants at a much lower cost than doing 30x sequencing. We hope that this combination of cost-effective sequencing and cloud-based genomics can be a contribution towards democratizing genomics research. And further, we look forward to novel discoveries and development of novel therapeutics to improve global health ultimately. And with that, I want to thank you for your attention. I want to thank AWS for the opportunity to speak and for the great platform and service and all the help that they've provided, especially in the early days. Thank you also to Prevo, who helped us set up some of our AWS Batch infrastructure and Nextflow Tower. Thank you to all our collaborators around the world and to the whole Variant Bio team, especially Tyler Stanley, who was our brilliant cloud architect.